Welcome to our second edition of our panel discussion, Stimulating Growth, Cultivating Solutions by Eppendorf. Our second topic today is stem cell in research and clinical applications. I'm Stephan Goldenbaum, your moderator for today, and I also warmly welcome our three discussion partners here in the panel. Dr. Philipp Nold, stem cell expert at Eppendorf, welcome. Also with us, Dr. Marcia Mata from the Cell and Gene Therapy at the Catapult in the UK, and also welcome Professor Bernd Giebel, leading a working group at the Institute for Transfusion Medicine at the University Hospital in Essen. Welcome all together. Philipp, you were involved in your research with stem cells and working or developing also a therapeutic approach. Can you give us an overview about the current systems and the situation in this field?
Sure. Thanks for this question. Actually, it's a very exciting situation that we are right now in. So we are facing the transition from a 2D based approach into the bioreactor systems, if they are, for example, oscillating plates or multi-layer stacks, or if they are hollow fiber system, stirred tank systems. Though we are at the point right now where the field is seeing the demand for more cells. And as they are in 2D systems, they are getting to this laborious work to achieve these numbers of cells. So they are switching to controlled systems right now to achieve the cell numbers they are needed for preclinical, clinical studies, and also for production in stem cell fields.
You already touched the field of stem cell therapy. What are the main stem cell therapies at the moment, and what are the main areas of their applications?
So maybe we look back a little bit. So the first stem cell application actually took place in the last century, in 1956, when bone marrow was transplanted into a human to cure leukemia. So in that time, and still in these times, the hematopoietic stem cells are used as a cell replacement. Mm-hmm. But since that time, a lot of things happened and new things were discovered. So these days, the mesenchymal stem cell, the multipotent stem cells, are used for their immunomodulation properties. For example, in 2008, Katharina Leblanc used these cells in a GVHD, in a graft versus host disease, to treat a child. And since 2006, when we look back to this year, when Yamanaka published his extraordinary work in Nature regarding the induced pluripotent stem cells, the iPSC, a new field opened up in form of organ replacement, tissue regeneration.
Mm-hmm. So also different kind of stem cell types are used for different kind of applications.
Correct, because stem cells, we use stem cells for their potency to differentiate into specific cell types. So as I said, induced pluripotent stem cells are more potent, for example, than mesenchymal or hematopoietic stem cells. They are multipotent.
So what are the current major fields in stem cell research you can describe?
So right now we see the phase where the cells are used as a cell therapeuticum. Mm-hmm. So we are using, as I said, the hematopoietic stem cell that we are doing since years, mm-hmm. the mesenchymal stem cells in the field for regenerative therapeutics, for example, repairing knee joints, but also using them in the immunomodulatory settings that I already described in the GVHD, for example. And since a few years, there's also new discoveries made that the mesenchymal stem cells, not acting the cell itself, but their secretome is working on that. I'm pretty sure Bernd will talk about this in more detail. And for sure, the iPSC field, using all the potential to generate cells of interest for organ replacement in future and also tissue repairing.
That represents what you described here, a little bit the present state. Where would you see future trends coming up, or what are the main trends in the future when we look into stem cell research, from your point of view?
So looking into the future, and as I said, we need more cells and we need more well-characterized and quality cells. We are now looking into having cells that are made in a standardized manner, where the protocols producing those cells are aligned and harmonized to compare it between the different groups to have a high quality product in the end, with the help of the system, using automation and also therefore reducing the risk of potential human failure. Risk mitigation is one buzzword that comes into my mind. So we are just now really, again, and that sums it up a little bit, in the translation from the 2D into the 3D systems.
Okay. That's a very interesting point you bring up here, which also leads me directly to our next guest here, Dr. Marcia Mata. Marcia, you are actually working intensively with 3D expansion models, using different kind of stem cell types to develop a therapeutic approach. What are you doing in a nutshell? Can you give us an overview?
So our aim is to establish a scalable platform for the delivery of in this pluripotent stem cell product. And as Dr. Philipp already mentioned, one of the main challenges is actually have a standardization on the product delivery, as well as increase product yield and of course decrease costs. So this is our main aim, and to establish a GMP platform from end to end, since expansion, differentiation until fill and finish.
And could you describe for our audience quickly what are 3D expansion models?
So a 3D expansion model is very actually different of what is widely known in the academics by the 3D organoids. So here what we are trying to achieve is the expansion of pluripotent stem cells. So we seed the cells into a stirred tank bioreactor as single cells, and they will aggregate and form these aggregates and expand like that. So then we can control the different process characteristics, such as aggregate size and feeding strategies to this automated platform, as well as searching for optimization of the process, such as seeding density and stirring speed.
How would you describe the challenges that need to be addressed for the requirements developing a therapeutic approach?
There are several challenges, as you mentioned. One of them is that there is one process that will fit all. So it will be very dependent on the product that you are trying to achieve, because it depends on the cell dose as well as the platform that you're culturing your cells, either 2D and 3D. So as Philipp mentioned, there is an increase on trying to move into 3D due to cell productivity and yield, and also to decrease the costs. But again, it will be very dependent. When scaling up, for instance, in the hematopoietic differentiation field, there is a need for cell selection. But nowadays there is a gap in technology that will actually allow for the large number selection under GMP and integrated with the process. And finally, the last thing that I wanted to mention is about starting material. So it has been shown that depending on the cell line, you will have different results. So certain cell lines are more prone to differentiate into the different products. So there is not a standardization here, and it's actually quite important to try to further characterize our products and to fully understand our process.
Okay. You spoke about cost efficiency. You spoke also about the performance of different cell lines and touched the topic of scaling up. Do you see a general trend for up or outscaling in the area of stem cell research?
So again, it will depend a lot on your process and actually the technology that is available. So of course we want to try to make our process as cost effective as possible, so it can be actually delivered to the patients. So for instance, in the cardiomyocyte field, we see a trend. We would like to see a trend of scaling up, because that will be the most cost efficient. But nowadays, what has been mostly out there is still culture under 2D. So so we can use hollow fibers, for instance, and that will be more of a scaling out approach. But again, it will depend on your indication and your process.
Thank you, Marcia. You have also mentioned the regulatory aspects previously, and stem cell research is and was always discussed very, very controversial. How does the regulatory landscape look like from your opinion?
So this is a very exciting field, but still very novel. So there is a lot of questions that still need to be answered. And in terms of regulatory, one of the major things that I foresee is making sure that we don't have any pluripotent stem cell or actively dividing cell in our final product. Of course, this will be challenging because it also means that we will need assays with a really high sensitivity for this product. Or for instance, there has been clinical trials where they had to perform whole genome sequencing. But again, it's quite challenging because it's a lot of data, and also it's difficult sometimes to actually interpret it.
Thank you, Marcia, giving us insight into your research. Our next guest is a leading expert in research with extracellular vesicles and discovered their potential for a medical benefit. Welcome, Bernd. To start with, which are the most common stem cell therapies you can describe?
Thank you for inviting me and having the chance to discuss with you this exciting topic. And I think Philipp already mentioned that there are different types of application, and at first it's the cell replacement strategy, which for example in hematopoietic stem cell research is the case, that you transplant hematopoietic stem cells, they engraft into the bone marrow of the recipient, and they form hematopoiesis all the life long. Then there are a couple of other diseases where the tissue degenerates, and it was thought that you transplant stem cells and that they also migrate into these defect tissues and replace lost cells. And one cell approach, which also Philipp mentioned previously, are the mesenchymal stem cells. And originally they were also considered maybe to be some type of pluripotent. So in the beginning of the millennium, there were reports like brain into blood and blood into brain, and they suggested that somatic stem cells have capabilities to transdifferentiate, to migrate into the tissues, to transdifferentiate into the cells which need to be replaced and to form these cells. And since there are acute diseases like myocardial infarction or ischemic stroke, people had to think of off-the-shelf products. That means that cannot be the autologous cells. That means that should be from foreign allogenic donors. And with that, immune therapy comes into immunity comes into play. Because if you transplant something foreign, it's rejected. And that has also been mentioned by Philipp. It was found that mesenchymal stem cells do suppress immune responses, and with that MSCs are applied in more than 1,000 clinical trials until now, either for cell replacement or for immune therapy. And then of course, what Marcia talked about, there is these IPS things. So it's the questionable whether MSCs are indeed pluripotent. Apparently they are not, but the real pluripotent cells are very interesting because they can transdifferentiate, and we need to learn to address the challenges you discussed already with Marcia.
Thank you, Bernd. You spoke a lot about stem cells and stem cell therapies. Now you have specialized to a very, very new field with a completely different approach. Your research is focusing on extracellular vesicles. Could you give us an introduction into extracellular vesicles and what they are doing?
Yeah, so the interesting thing with the mesenchymal stem cell was that people started looking where do the cells migrate to, and they tried to recover them in affected tissues, which apparently benefited of the mesenchymal stem or stromal cell therapy. And then it turned out that these stem cells are hardly discovered in the affected tissue, and most end up in the lung and disappear within a few days. And with that, it came across in the middle, so 2005, 2006, that eventually MSCs do not work in cell replacement, but in a paracrine mode of action. To that end, people wondered whether if they act paracrinely, whether they do secrete these therapeutic activities into the supernatant if you raise these cells in culture. And indeed, it had been shown that if you apply conditioned media instead of the cells, you receive, some researchers observed comparable effects.
So the extracellular vesicles are actually a more or less a side product of stem cell research.
Yes, they are a certain type of side product. So what I was going to discuss is that the people were searching for the activity in the conditioned media, and then they fractioned the media, and then they recovered the activity in by filtration in a fraction larger than 1000 kilodalton, or which could be pelleted by ultracentrifugation, and scanning electron microscopy showed that there were many vesicles, and extracellular vesicles appeared as exosomes in 1983, and in 1996 it was clear that they are not just transporting debris, but that they have functional activities, and today we think they are part of an ancient communication system, that each cell can release extracellular vesicles which are assembled in cell-specific manners, and that they can be targeted to selected target genes to have multi-signaling to these target cells, and that eventually viruses have captured the machinery. So our knowledge from viruses, what we have, how they can spread information, maybe that is an effect of what we today think extracellular vesicles do. And that is if they are now the agents which execute the stem cells activity, they have, of course, several advantages to be applied.
I see. Thank you. And when you describe the work and the preparation of the use of extracellular vesicles, what kind of advantages and benefits do you see in working with extracellular vesicles to create a therapeutic approach?
Yeah, so the big advantage is, that is what Marcia already discussed, is if you have pluripotent cell product, you need to be sure that there is no single cell which is pluripotent left, otherwise you have to expect teratoma formation. And that is also, even though if MSCs appear safe, you may apply them also to little children, and you never know what is the capability of eventually cells which survive. Will they have tumor formation potentials? And therefore you always have a risk if you transplant cells with a certain self-renewal potential that tumors can be formed at a certain time. If these are that small, they are 100 to 150 nanometers, at least those which we like to use or which we used, and they can be sterilized by filtration. They have no endogenous self-renewal potential, so they have no endogenous tumor formation potential. The overall handling is much more simple, and in animal studies we learned that they are, in an ischemic stroke model, that they are indeed as effective as the paternal MSCs they were harvested from their supernatants.
Thank you, Bernd. And where do you see the major challenges for a realization of a finalized therapy here using extracellular vesicles?
Yes, indeed, there are several challenges, and one major challenge is inherited from the MSC field, and that is that MSCs are a very heterogeneous cell entity. So depending on the donor and on the passage or preparation, not all MSCs are apparently therapeutically active, and that has resulted in these controversies whether MSCs are a potent therapeutic agent or not. And this heterogeneity is indeed inherited to the EV field, and we learned that depending on the donor and passage and culture conditions, some of the MSC EV preparations which we prepare are considered to be active, and others do not provide this suggested therapeutic activity. And therefore, it's a challenge to identify functional readouts which allow us to identify the right preparations which we can apply from those which we should not apply. Then the scalability for sure is a big challenge, and so we would not like to produce products which can just be applied to a few handful of patients, but maybe to a broader patient number. And the big advantage in the MSC field which we have is that we can use immortalized MSC lines, and that is what we have recently performed. And of course we now need to learn whether the EVs released by these cells provide the same potency and do not induce any additional risk. We do not see any risk of the immortalization process, so I think that is very promising. And after that, then we can use upstream and downstream processing, which need to be optimized. So that means these potentially immortalized MSCV producers, they can be expanded in high numbers in bioreactor, and we have to identify the best bioreactor type, the media, the conditions of the bioreactor tank, and to create a huge amount of conditioned medium, which will be the basis for extracting the EVs in further downstream processing. And indeed, there are a number of techniques available, such as tangential flow filtration, which would allow us to really extract the EVs from giant volumes. And of course, each of the production steps, whether it's upstream or whether it's downstream, it may affect the activity of the resulting EVs. And thus, we always need to test if we change any parameter in the production chain, whether it affects the activity. But we are very optimistic that we can manage to have these production processes optimized, and a big advantage compared to cells, we can sterilize the product by filtration, and we think that there are also really potent methods to make it a really broadly applied off-the-shelf product.
Thank you, Bernd, for sharing your research with us.
Yeah, it's my pleasure, and thank you very much for providing me the chance to introduce the MSc field.
Thank you. We have learned today a lot about stem cell research, different forms of therapies. We also got insight into very specific fields like 3D expansion models, and as we just have heard, also the work with extracellular vesicles. I thank also my guests today. Philipp, thank you for being here. Thank you, Marcia, also sharing with us your work at the Catapult network. Thank you also, Bernd, giving us insight into your work with the extracellular vesicles. Visit our website to learn more about bioprocess for stem cell research and other applications. We are looking forward to welcome you for our future discussions. Thank you.